Good morning, good afternoon, and good evening to everyone attending today's Journal Club from the different parts of the globe. Our today's Journal Club is sponsored by the American Thoracic Society's Assembly on Allergy, Immunology, and Inflammation. My name is Anukul Shenoy. I'm an assistant professor of microbiology and immunology and pulmonary and critical care medicine at the University of Michigan Medical School at Ann Arbor. And today I'll be moderating the discussion on the paper titled Prevention of Respiratory Virus Transmission by Resident Memory CD8 T cells by Sarah Michaleks and Dr. Jacob Kohlmeier and colleagues that appear in the February 2024 issue of Nature. Joining us for today's introduction and the discussion of the presentation is the lead author, Dr. Jacob Kohlmeier, PhD, Associate Professor, Microbiology and Immunology at Emory University School of Medicine, and two expert discussants, Dr. Ellen Foxman, MD, PhD, Associate Professor of Laboratory Medicine and Immunobiology at the Yale School of Medicine, and Dr. Luisa Morales Nebreda, MD, Assistant Professor at the Division of Pulmonary and Critical Care Medicine and the Center for Human Immunobiology at the Northwestern University Feinberg School of Medicine. And finally, here to give us the presentation along with Dr. Kohlmeier is Ms. Sarah Michaleks, who is a PhD candidate in the Immunology and Molecular Pathogenesis graduate program at the Emory University. If you have any questions for the presenter or discussants at any time during the webinar, you can always use the chat function in the Zoom and enter your question as a message. I will raise these questions during the question and answer session after the presentation in the order of which they appear. We anticipate we will get through all of your questions, but if some questions do go unanswered or if time runs out, please feel free to reach out to the authors with your questions, and I'm very sure they'll be more than happy to answer those questions by email after the webinar. So with that, I will let Ms. Sarah Michaleks begin the presentation. Sarah, the floor is all yours.
Right. All right. I'm going to start just briefly here. I'm filling in for Ida Udbach, who is Sarah's co-first author who couldn't join us today. So I'm going to begin the presentation just with a brief introduction and talk about figure one. And then I'm going to turn it over to Sarah so that she can actually talk about the work that she actually performed in the lab. It's part of this manuscript. So thanks again for the invitation. We're really excited to be here. So I'm going to start with a little bit of brief background. Maybe. Yes. All right. So why are we interested in the role that T cells could potentially play in protecting against respiratory virus infection? So this is a slide that I'm sure many people on here are familiar with. This is the CDC's study looking at influenza vaccine effectiveness over the last 15 or so years. And what you can see looking at this is that the way that they're judging effectiveness here is never really gets much higher than 60%. So even though vaccines are, I'm not saying don't take your vaccines, by all means, get your vaccines. But as far as their overall effectiveness, there is still a certain degree to which these vaccines fail to provide protective immunity against circulating respiratory virus strains. And this varies some from year to year. And so, we've seen this with influenza. We've certainly have all seen it, firsthand in the news and in the scientific literature with the SARS-CoV-2 infection, where as viruses encounter this antibody-mediated immune pressure, they can rapidly mutate, and these mutations can then help them avoid some of this pre-existing antibody, allowing virus to transmit on to the next individual and the infection to continue to spread within the population. So our group has had a longstanding interest in trying to look at what T cells might be able to contribute to protective immunity against respiratory viruses. And so just to kind of demonstrate this in graphic form, one of the key components in which T cells can help assist with protection is that as viruses mutate in response to an initial antibody-mediated pressure, changing these epitopes on these surface proteins, making it so that antibody cannot bind as well or be as advantageous, a lot of other epitopes that the virus encodes for on these internal proteins are not under the same degree of antibody-mediated pressure. And many epitopes that T cells can recognize are found within these internal viral proteins. And these proteins are often highly conserved across different strains of the same virus. So a T cell that could recognize an epitope in the original virus, in all likelihood, that same epitope will be present in a mutated virus because these internal proteins are not prone to undergo near as much mutation. So one possibility is that having T cells that can recognize these internal epitopes can provide cross-protective immunity across a number of strains for a particular virus. And so our lab and many others have been interested in tackling this question and working on it for the last several decades. And just to give a little bit more introduction into memory T cells themselves. So this is a very simple schematic looking at what happens with the dynamics of virus-specific T cell development after an infection or vaccination. You start, of course, with a very small pool of naive T cells specific for a particular epitope. Following infection, those numbers expand. Virus is then cleared, leading to contraction of the majority of that virus-specific population, but a small fraction, 5 to 10%, then persist long-term as memory T cells at a much higher frequency than their naive precursors. And these cells have undergone a number of transcriptional and epigenetic changes such that they're able to respond rapidly with immediate effector function should they encounter their antigen a second time. So it doesn't quite require the same lag time that ramping up the naive T cell response does. And so the potential for these memory T cells, we believe, is fairly high as far as their contributions to antiviral immunity. But it's not just any old memory T cell that we're really interested in. We're particularly focused on a unique population of T cells, first described by Dave Mazepust and others, known as tissue-resident T cells. So as the name implies, these cells remain permanently in peripheral sites or within internal organs. So unlike central memory or effector memory T cells, they do not recirculate throughout the body, at least not in large numbers. And that allows them the ability to be uniquely situated such that following initial infection events or initial transmission events, these memory T cells are located in close association with those targets that become infected during those initial infection events. And their ability to mediate fast effector functions such as cytokine release or cell lysis holds the potential for these cells to rapidly eliminate an infection before the virus can propagate and spread, leading to pathology. And so these cells canonically have been defined through various mechanisms. We use approaches such as intravital labeling so that we can discern between memory T cells in the bloodstream versus those in the tissue. TRM cells also have this unique phenotype where they express molecules such as CD103 and CD69, both of which contribute to their maintenance within the tissue and prevent them leaving the tissue and re-entering the circulation. And dating all the way back to Walter Gerhardt's work at UPenn 30, 40 years ago, it's been well characterized that these cells, when present, can provide heterosubtypic protection against influenza virus challenge. We started out this project with the idea that the way we do our experiments in animals when it comes to respiratory infections is just not very physiologic, right? And we all do the same thing. We still do this in the lab too. We suspend usually a pretty good amount of virus in liquid. We anesthetize an animal and we add it dropwise into the nose or potentially via aerosol to these animals. It's a very great method for delivering an exact amount of virus at an exact amount of time, but it's also not how viruses spread around in nature, or respiratory viruses at least. And so we wanted to see if we could try to develop systems that would allow us to ask this question of, if we generate tissue-resident memory T cells under a physiologic setting of natural virus transmission, where it's likely that the infection is seeded only by the transmission of just several virions that take hold and then propagate and spread the infections, could memory CD8 T cells in the respiratory tract actually efficiently surveil that large space under that kind of scenario and prevent infection following natural respiratory virus transmission. So that actually sounds like it was a question we were interested in. It's not quite as easy to tackle as it might have seemed at first, mostly because a lot of the models we use in animals, such as mouse-adapted influenza viruses, this is true also for mouse-adapted SARS-CoV-2 viruses now, they don't actually transmit between wild-type animals. There are a few models where transmission can occur, neonatal mouse models and a few others, but none that really lended themselves well to being able to ask mechanistic immunological questions. There are other good animal models for viral transmission, guinea pigs, ferrets, Syrian hamsters with SARS-CoV-2, but anyone who works in these models knows that the immunological tools available to play with are limited, to put it nicely. And so we settled back on a virus that I worked with some as a postdoc, and that is Sendai virus, which is a mouse para-influenza virus, very similar to PIV1 in humans. And unlike the mouse-adapted flu or SARS-CoV-2, this is a natural mouse pathogen. They screen for it in all your vivariums because they don't want it because it spreads around like crazy. But it does naturally transmit in mice via both contact and aerosol routes. And we knew some information about the virus itself as far as the immune response in Black 6 mice. We know the immunodominant T cell epitopes, for example. So with that in mind, we set about building or borrowing a few tools that we could try to use to look at transmission. And the first of this was a Sendai virus that encodes luciferase. So this allows us to longitudinally track viral infection between mice within a cage in a sort of high throughput, not super invasive manner. And in a manner in which all of my students and postdocs wouldn't quit for doing a million, plaque assays. So Charlie Russell at St. Jude was kind enough to provide us with this luciferase Sendai virus. This is an example of what it looks like with the imaging. And then we can calculate the total flux in photons per second over the course of an infection, and we get a nice looking infection curve. And then importantly, in some mice, we did correlate the actual viral titers with the bioluminescence that we saw by the virus, and there's a very strong correlation between those. So now we have a tool to longitudinally look at viral spread. And the next issue became developing a way to generate Sendai-specific TRM or CD8 T cells that recognize the Sendai virus without generating either CD4-specific Sendai cells or antibodies against Sendai virus itself. And so thankfully, we have a number of great colleagues with Jan Christensen at University of Copenhagen, Anise Lowen, a terrific influenza virologist down the hall. And we had the knowledge going in of the 9-amino acid immunodominant CD8 T cell epitope for Sendai virus. And so working with Jan, we developed a series of adenoviral vectors that are replication deficient that express this Sendai virus epitope. Working with Anise, we built that 9-amino acid Sendai epitope into a number of influenza virus vectors, including live attenuated vaccine versions. And so now we could immunize animals and then generate Sendai-specific T cells without generating Sendai-specific antibody or Sendai-specific CD4 T cells, allowing us to ask questions specifically about what CD8 TRM could do. Okay, so I'm just going to briefly kind of touch on some of our approaches here for immunizing animals. I'm not going to show all of the data, but certainly I'm happy to answer any questions, or Sarah can answer questions about it later. So this is kind of a basic experimental setup that we use. So the PR8 here, this is an influenza virus. And in two cases here, we're using an influenza virus where it says SenNP. This is where we've knocked in the 9-amino acid Sendai CD8 T cell epitope into the neuraminidase stalk of that virus. And then we can immunize animals in two ways. We published on this previously, and I'll explain why we do it the two different ways. So this is the IN here is just intranasal infection with this flu virus. This generates Sendai-specific CD8 T cells, both systemically and resident within the respiratory tract. We had published several years before that if we use this same virus at a much higher dose, but go in via a non-respiratory route, either intramuscular or intraperitoneally, which is what we did here, IP, we get an abortive infection with the virus. So we get one round of replication, but that's enough for us to generate a Sendai-specific response to that send-NP epitope. But the key is, going in IP here, we can generate similar numbers of circulating Sendai memory to an intranasal infection, but we do not generate hardly any TRM within the respiratory tract. So this sets up three scenarios that allows us to look at transmission. No Sendai-specific T cells using the wild-type virus, circulating Sendai memory T cells using the PR8-SNMP IP, or circulating and resident memory T cells when we do an intranasal priming. And so just to make sure this worked like we thought it would, we use this to immunize groups of mice. We wait 5 weeks in this case. And then in this case, we're not looking at transmission. We're just directly infecting them with Sendai luciferase and looking to see what happens to viral kinetics over that time. And so this is a complicated, bit of data. I'm just showing you all of the data here. Each dot here is nasal lavage from an individual mouse over time. But the take-home message of the curves is what I want you to focus in on. And so if mice have Sendai-specific TRM here in this purple group, you can see they have significantly lower viral titers as we measured by nasal lavage compared to either circulating memory or no Sendai memory. And when we do it by IVUS, we see the same thing here. So if you have Sendai-specific TRM, you get decreased viral flux or total amount of bioluminescence from the virus, and the virus is cleared faster compared to having either just circulating alone or no Sendai memory. And that's quantitated here on the right with the area under the curve, where each symbol is an individual mouse here. So essentially, this was just kind of a really fancy way to show what everyone else had shown for 20 or so years. And so with this in mind, the idea that the system works the way we would have expected with a direct infection, we wanted to start to ask questions about transmission. And so there's two different ways we were curious about looking at transmission. One is, if you have Sendai-specific TRM, how does that impact your ability to transmit virus to a naive contact? Or if you have Sendai TRM, how does that protect you from becoming infected should you encounter an infected contact? So I'm going to tell you about the former here first, which is, how does it impact your ability to transmit virus to a naive contact? And so we did the same sort of experimental setup with these primings, waited 35 days, and again infected these mice with Sendai luciferase. And then at various times after this Sendai luciferase infection, either 1, 3, 5, 7 days, we took one of these mice and put it into a cage with 4 naive contact mice. And then what we're measuring here is the bioluminescence of these contact mice. So do they become infected? And just to sort of sum up the data here on the bottom, what you see is that if, 1 day later, if you add these infected mice in, if you have Sendai TRM, you still get a little bit of transmission, but it's certainly nowhere near as much transmission if you have no Sendai-specific T cells or circulating only Sendai-specific T cells. But by day 3 now, if you had Sendai TRM, you're no longer transmitting at all to any of these naive contacts. Day 5, the circulating memory now no longer transmits. So circulating memory is contributing something, but it takes a while for that response to ramp up and be recruited into the lungs where it can help eliminate virus. By day 7, nobody's transmitting. So, I think not surprisingly, these data showed us that having those TRM present, which we know is decreasing virus faster and to a lower level, that's also significantly shrinking that window in which they can transmit virus to naive contacts. So at this point, I'm going to turn it over to Sarah, and she's going to take you the rest of the way home. Yeah, or you can just move up a seat. There you go.
Sure. All right, and so the data that Jake just showed was looking at the scenario where the index mouse has been immunized. But as he mentioned, we also wanted to look at how TRM present in the contact mice could prevent them from becoming infected. And essentially, having those TRM present throughout the respiratory tract, we're able to survey it and rapidly eliminate any initial infection events. And so here's our experimental schematic for this experiment. It's very similar to what Jake just showed, but now we have the contact animals that are being immunized with our PR8 wild type, so no Sendai virus specific immunity, and then our PR8 Sendai viruses either intraperitoneally or intranasally. And so after 35 days, we took a separate mouse, that would be our index mouse, infected that mouse with Sendai luciferase, and then dropped it into the cage with the other contact mice and monitored by in vivo imaging the transmission from that index mouse to the contact mice. And so looking in panel B here, what you're seeing, these are the bioluminescent curves from those groups, where this dotted line at 6 logs is representative of our threshold for infection. So any animal whose curve goes above that 6 logs, we consider that animal to have become infected. And then in panel C and D are looking at the ways we quantitate this data. So in C, we have, as Jake mentioned, the area under the curve. So this is essentially answering the question that if an animal becomes infected, how does having the presence of those TRM or circulating memory T cells decrease or impact the total viral burden of that animal? And so what you can see here in our purple group is that there were only a couple of breakthrough infections that did occur, but those that did had a significant reduction in the area under the curve compared to those that had circulating memory alone or lacked any Sendai virus specific immunity. And then in panel D, this is looking at the probability of infection. So the proportion of animals in that group that became infected. And you can see the reduction is pretty dramatic. And so another important point to point out with this data is that we also took separate cohorts of mice that we immunized and took them down at the time of co-housing after 35 days to look at the number of TRM that were present in the 3 main respiratory tissue sites we're interested in, the bowel, the bronchoalveolar lavage, the lung, and the nasal cavity. And so what you can see here is that when we look at the correlation between area under the curve and the probability of infection for animals with that number of TRM, we can see a strong negative correlation in all these tissues. And so this indicated to us that the number of these tissue resident memory CD8 T cells is really important for the level of protection against infection from transmission. And so there were a couple of key questions that we wanted to ask using this model. So the first one is looking at how durable is this tissue resident memory T cell response against preventing transmission. And then the second question is about which effector mechanisms are really responsible for what we're seeing in this model. And so to answer the first question, because this is a pretty brief explanation, we used the model I previously described where we immunized mice with some various vectors intranasally. So here I just have used an X31 wild type as a positive control. This is an H3N2 influenza virus, and essentially no animal in this group has any Sendai virus specific immunity, so we expect every animal to become infected. And then our PR8 Sendai virus and X31 that also has that immunodominant CD8 T cell epitope in there. And finally, we have our, that Jake alluded to earlier, our replication deficient adenoviral vector. And so we immunized these mice, waited at around 6 months, and then co-housed them with an index mouse and measured transmission. And so in B, what you're looking at are the bioluminescent curves. And you can see pretty clearly that there's a, the level of protection against transmission varies between the different groups. But in particular, our adenoviral vector was largely protective. There's one animal that had a very slight breakthrough infection there. And so we can see these results reflected in the area under the curve, as well as the probability of infection, where this group that received, in particular, adenoviral vector immunization were protected for at least 6 months and likely longer than that we would expect. And so to kind of explain the differences between the different vectors that we saw in this experiment, these are looking at the number of TRM in these tissue sites for these 3 different vectors after 6 months, as opposed to 1 month. And so what you can see is that the number of these TRM for our PR8 Sendai virus and our X31 Sendai virus are significantly lower in all the tissues we examined. So, and in corroborating our correlation plots, as you can see, the number of TRM that you have present is important for the ability to protect against transmission. And so the second question, which is perhaps a little bit more interesting, is that we wanted to look at the effector mechanisms of these tissue resident memory T cells. And so the first experiment that we did is we actually sorted antigen-specific T cells, Sendai virus-specific T cells from tissues in the bowel, the lung, and the spleen, and also at 3 days after. This is just a direct infection challenge, and we performed bulk RNA sequencing. And so what particularly can draw your attention to in panel B is that these TRM, after they're activated, so this is our purple group and our dark blue group here. What you can see is that they've upregulated genes associated with effector function, such as interferon gamma, granzyme B, perforin, and some other chemokines. And so, however, we don't see this increase when we're looking at the effector memory that have been activated in the especially in the lung. And so these are kind of some of the effector mechanisms that we wanted to focus on. And so this is the first experiment using the first model that we talked about, where the index mouse is being primed with these tissue resident memory T cells. So we took wild type mice, of course, as a control, perforin knockout mice. So they don't mediate cytolytic activity by the perforin granzyme mediated cell death pathways, and interferon gamma receptor knockout mice. So mice that cannot respond to interferon gamma. So we took these mice, we immunized them with a live attenuated influenza vector with that immunodominant CD8 T cell epitope to account for any differences in pathology that we may see with our PR8 Sendai vectors. The PR8 Sendai vector can cause significant to the pathology mice, but the live attenuated vector does not. So that's why we chose to use this with the knockout mice. And then we, after 35 days, infected them directly with Sendai luciferase, and then put them in the cage 3 days later with naive Black 6 contact mice and performed in vivo imaging. And so these curves in F are looking at the naive contact mice, not the index mice. And so what you can see here is that in our wild type group, so these animals had the TRM, there was one breakthrough infection out of the whole group that happened. Whereas with our perforin mice, now we're getting a few more breakthrough infections, but not very many. And then with our interferon gamma receptor knockout mice, over 50% of the animals are becoming infected. And so you can see this reflected by the probability of transmission, where the interferon gamma receptor knockout mice were significantly predisposed, or the contact mice that were co-housed with the interferon gamma receptor knockout mice were significantly predisposed to becoming infected. And so we also wanted to confirm these results using the other model that I mentioned. So now we're taking the contact mice and we're priming them again with our live attenuated influenza virus vector with that 9 amino acid epitope insertion. And here we have wild type mice, perforin knockout mice, as well as both interferon gamma knockout and interferon gamma receptor knockout. And we have a separate group that we gave a wild type mice that we gave a fast ligand blocking antibody to during the co-housing and transmission period. As many may know, the fast knockout mice have a pretty significant lymphoproliferative disorder. So this is the way that we chose to go about this. And so we took these mice, co-housed them, performed in vivo imaging, and here there's a lot of curves. What you're seeing are these bioluminescent curves here. And so just to walk you through it, what you can see is in this top left plot with the red group, as expected, every animal has become infected without any Cinnavirus-specific immunity. In our purple group, these are our wild type mice. We are having a few breakthrough infections. But in the bottom, when we look at here in the green and the yellow, our perforin knockout group and our fast blocked group, what you can see is, again, there are a few breakthrough infections, but just a handful, whereas our interferon gamma knockout group and interferon gamma receptor knockout groups, virtually every mouse is becoming infected. And so you can see the same results here in the quantitation. In particular, looking at our pink and our blue groups, the interferon gamma knockout mice, we see that is statistically, they have an increase in their probability of infection in area under the curve compared to the purple group here, which is our wild type mice. And so this suggested to us that interferon gamma secretion seemed to be a dominant mechanism by which the CD8 tissue resident memory T cells were able to limit viral transmission. And so we wanted to dig a little bit deeper into this mechanism of what interferon gamma from these T cells is doing. And one of the challenges that we've had with doing this is that if we just take mice in our transmission model and perform RNA sequencing on them, we can't tell specifically which changes are due to interferon gamma versus which changes are occurring because the animals are just becoming infected. And so to do this, to get around this, we actually used a peptide dosing model. So we prime mice with our live attenuated influenza virus vector. And then after 30 days, dose them intranasally 3 times with a Cinnavirus nucleoprotein peptide to virtually activate all of the antigen-specific T cells that are present in the respiratory tract. And we use both wild type and interferon gamma knockout mice to ensure any differences we saw are truly a result of interferon gamma. And then we FACS sorted nasal cavity epithelial cells at 30 days without peptide dosing, and then 3 days after the peptide dosing. And so we did bulk RNA sequencing on that. And what you're seeing here on these PCA plots, what you can appreciate is that our green and our peach colored group here, these are the epithelial cells at the resting memory time point. They're clustering together. And now after we've dosed them with peptide, they're clustering very distinctly. And when we look at some of the DEGs here, we can see our purple group here in this bottom bar. These are our wild type animals that have been dosed with peptide. And in these animals, we're seeing significant increase in transcriptional programs associated with antigen presentation, as well as some interferon stimulated genes. And this increase in these DEGs is lost now when you're looking at the interferon gamma knockout mice. And so we also wanted to look not only transcriptionally, but also at the protein level about some of those DEGs we were seeing. And so I had a panel with a variety of different activation markers, but the one that really stood out, as you may expect, would be MHC class 1 and 2 presentation. So here you're just looking at histograms and in-F flow cytometry data. So you can see that the increase in these MHC class 1 high epithelial cells and MHC class 2 positive epithelial cells, it's a pretty dramatic increase after the peptide dosing that is completely lost when interferon gamma production is no longer present. So this also showed us that interferon gamma signaling from these TRM is increasing the antigen presentation on these nasal cavity epithelial cells and initiating that antiviral response to prevent transmission. And so finally, we took supernatants from the nasal cavities of these mice and performed a multiplex cytokine and chemokine assay. And so what we saw, this is a nice positive control, is that interferon gamma production was indeed increased in our wild type mice after stimulation. And in those mice, we also saw an increase in interferon gamma induced chemokine secretion, CXCL9 and CXCL10, to help recruit other lymphocytes and leukocytes to that nasal cavity tissue. And in this SPAR panel, so we did see some cytokines and chemokines that were increased in both groups after the T cells were activated, but it was not significantly different between the interferon gamma knockout in the wild type mice. And so just to conclude some of this data that we've shown you, and we've gone through it pretty quickly, but these mucosal T-cell based vaccines, particularly ones that induce these tissue resident memory T-cell populations, are able to limit respiratory virus transmission, not only from an immunized individual to a naive individual, but also prevent a immunized individual from becoming infected themselves. And so this would really serve to limit respiratory virus transmission in the population level by both of these means. And so I've also shown you that this protection against transmission can be durable, particularly with our adenoviral vector immunization strategy. And we've also seen that interferon gamma that's being secreted by these CD8 tissue resident memory T cells is able to limit transmission by inducing antiviral gene transcriptional programs in these in epithelial cells, as well as increasing antigen presentation and other lymphocyte attracting chemokines to recruit other cells to the site of transmitted virus. And so with that, we'd like to thank everybody in the lab, and particularly Ida Udbak, the other co-author on this paper, who's not able to be present today. And we'd like to take any and all questions you have, and if we don't have time, feel free to email us afterwards.
Awesome. Thank you so much for the great presentation, Jake and Sarah. I would like to open up the conversation to our discussants as well as audience members. Just as a reminder, if any audience member has a question, please feel free to state so in the chat, and I will be happy to ask you to unmute at a specific time, and you can feel free to ask your question in person. And if you cannot unmute, feel free to type the question in the chat, and I'll be happy to read it out to Jake and Sarah or the other discussants. So with that, I'll leave it to Ellen and Louisa to ask questions and thoughts and share your ideas and stuff.
Sure. Well, I can kick it off. First, I want to say congratulations on this fantastic paper. It's really, really beautiful work. And I also love this model with the non-invasive monitoring of viral load that really enabled you to do this really interesting study. I have several questions for you, but one that my lab has been studying heterologous immunity, where the interferon response induced by one virus protects against other viruses. And I'm really curious, seeing this antigen experiment you talked about at the end, have you ever tried to see whether providing the peptide actually will protect against a different virus by inducing the interferon gamma?
Well, yes, we actually have done an experiment like that, slightly different from what you described, but we've actually immunized mice with a flu nucleoprotein expressing virus, so generating flu specific T cells and then dosing them with flu peptide, but challenging them with the Sendai virus transmission. And so
what happened? Oh, you're going to tell us, I guess. So.
Yeah, and so what we saw when we did that experiment is actually that the interferon gamma delays the onset of infection. So when they don't have any antigen specific for the challenge virus CD8s, of course they ultimately become infected. But we do see that it's substantially delays the onset of the infection.
Interesting. Wow. Yeah, that's very interesting. I mean, maybe with a less infectious virus than Sendai, it would prevent some of the infections. I wonder. Sounds like that's a very transmissible virus.
Interesting. Louisa, I saw you on mute. Yeah.
Really amazing work. I love the model, like Ellen said, very elegant. I just had a question. So, this is sort of known biology, but and I guess it makes sense to have focus on the epithelium, sort of looking at where interferon gamma receptor is expressed pretty ubiquitously. But one cell type that it's highly expressed is macrophages. So I wonder if you looked in your model, and there has been this description of T cells producing interferon gamma, macrophages sort of responding and having an interferon responsive transcriptional program, and then sort of forming these loops that continuously recruit cells to the lung space. So I wonder if you have looked at what happens to other cell types, immune cell types in your model.
So we didn't look specifically at how interferon gamma could impact like functionality of macrophages in the tissue. We did look at the influx of other innate immune cell types, such as we looked at inflammatory monocytes and NK cells into the tissue, and we saw that the influx of those cells was not significantly different when the TRM were present or not present.
Gotcha.
But yeah, looking at the functionality of the macrophages would be interesting for future studies.
I was wondering if I could ask a question. I mean, given that you brought in MHC and epithelial cells, I have to ask this question, right? I'm assuming that epithelial cells would have to present the antigens to the CD8 T cells to do this effector function. Is that something you've tested? The only reason I'm asking because people also have talked about bystander activation of TRMs and stuff.
Yes. Yeah, that's also a great question and something that we have thought about. So we started to answer that question actually using the NKX Cree mice that you've used in your Nature Comms paper. However, we bred up the mice crossed with a B2M flux, and we found that NKX is not actually expressed in the nasal cavity. So that approach did not work for us. And now we're going back and trying to do those experiments with a different Cree that is expressed for the nasal cavity.
Yeah, I think that's a really interesting question, Nicole. I mean, I think if I had to make like a prediction, I think epithelial cell presentation would be critical. Just because I think what the TRM are really doing here is stopping the virus propagating from those initial, small number of infected epithelial cells and preventing the spread. And so I'm curious about the cross presentation, but I think the kind of the timing of the way that would have to work would be after the virus has started to propagate, and then we would see the infection, right? And essentially we're wiping it out in some cases before we even can really tell there's an infection there. So my guess is it would be the epithelium, but we're like Sarah said, we've got some EpCAM Cree mice in now that we're crossing it to. We just haven't finished the experiments yet.
That's really interesting. I assume so also because, I mean, when you're giving your mice PR8 wild type, I'm assuming it's fair to assume that there are PR8 specific CD8 TRMs in the nose, right? And they are not protecting against Sendai. So right. I think it's fair to assume it's probably not the bystander activation. But I figured with MHC that I should ask this question. Yeah. I see a question in the audience from Edward Nirenberg. They have a comment, amazing work. I do agree that this convincingly supports a role for CD8 TRMs in limiting transmission. Two major questions come to my mind for me. As you note, epitopes of non-structural proteins tend to be stable. Given the extremely consistent effect of antibodies in driving escape mutations of the structural proteins across viruses, do you have concerns about what this suggests the viability of CD8 TRMs is in limiting infections?
Yeah, so I mean, it's a really interesting question, right? And all I can really, say about that without having any experimental data to back me up is that if you look at immunodominant epitopes for influenza, for example, in the human population, so if you look at like AO2 individuals and the matrix, I think it's a matrix 58 epitope, that has been present within influenza viruses in circulation for decades, even though it's a very strong immunodominant response. So there's certainly some evidence when you look back at viruses over decades where some epitopes might be there and then are lost, and as the virus evolves. But yet there's not strong evidence that immunodominant epitopes are under a lot of immune pressure that would drive their intentional evolution in a way that inhibits their ability to bind MHC or, something like that. So, I mean, my cautious answer is that I don't think there's strong evidence out there now that would suggest that, focused immunity on these epitopes would preferentially drive evolution.
Yeah. That also leads me to the other question they ask. I assume that I know the answer, but I'm going to let you answer it. Is do you believe your work excludes a role for humoral immunity in restricting transmission?
So
Absolutely
Yeah.
not. Right. So to me, the perfect vaccine is humoral immunity, right? So if we can vaccinate and make antibodies to which the virus can't escape, that is perfect. I just think that, for the most part, nature is smarter than we are, and the virus can often find a way around that. I guess the way I think about it is this is potentially a second line of defense, right? It can contribute to and strengthen the impact of humoral immunity. And where it might really have the potential to shine is in those instances where there is humoral immune pressure that pushes viruses to mutate in HA or something like that, that this is a secondary line of defense, right? That if it does escape pre-existing antibody and we're able to generate lots of TRM to a conserved epitope, that's just a backstop to humoral immunity.
Yeah, I for record, that was exactly what I would have thought you would say also. So, but I see Bruce has his hand up. Bruce, go ahead and unmute yourself.
Thank you. So it's a lovely presentation. I had a couple questions if we had a second. I've been impressed at how hard these antigen-specific TRMs to viruses are to find in humans. And I'm just wondering if you guys have thought about this, done experiments, or aware of sort of demonstrating similar protective mechanisms, even just to peptide in humans.
Yeah, so the lab, another student in the lab, that's actually her dissertation work. So the lab receives full human lungs that are relatively healthy, but were rejected for transplant for other reasons. And so we process those lungs, and she's done experiments looking at with peptides, looking at activation-induced marker assays and intracellular cytokine staining on those human lung cells. And she has seen the presence of those flu. She's looked at flu, RSV, and SARS-CoV-2, and we do see them present in the lungs, human lungs, and they also express CD103 and CD69, the two main tissue residency markers. If you have anything to
Yeah, I'm just going to add a little something to that, because I think it's a really interesting point. So when we started looking at a lot of our human lung samples, 5 years ago or so, I would have anticipated that essentially every person we looked at, their lungs would be full of TRMs because they've been exposed to these viruses over and over throughout their lifetime. And while what Sarah said is absolutely true, that we find these TRMs for various respiratory viruses in a number of people, there's another number of people where we don't find them. And we can, in some cases, find antigen virus-specific T cells for flu or other viruses that are not TRM. So they might just be circulating effector memory. And to me, the really interesting question that, it would be a heck of a long-term experiment to do, but try to get at this idea of, why aren't all people's lungs full of virus-specific TRM? And is there something where they decay over time if you don't see the virus enough? Are there other unrelated events that can cause TRM to either leave tissue or to die by apoptosis? I think those are really interesting questions. I just don't have a great answer, but I will say I was surprised at the number of people who didn't have virus-specific TRM in their lung.
But your data, though, it does suggest it doesn't last forever, right?
Yeah.
So I was interested if you could comment more on those kinetics. and also how does it relate to the kinetics of mucosal antibody persistence as well? Like if both were generated by a natural infection.
Do you want to?
Right, yeah.
Yeah.
So previous data from the lab has seen that, these TRM that are present in the lung and in the airways, they do decline over time. And we saw that also with some of my data. And we think it's largely due to, the presence of respiratory antigen. So in our adenoviral immunized group, those TRM are actually stable over time. And we've seen this is because some of those the adenoviral vector can incorporate into or establish antigen depots in alveolar macrophages that continues to kind of stimulate those TRM and give them, the threshold of activation signals that they need to remain and survive within the tissue. Yeah.
Well, why is it the whole virus or just some viral antigen, or is it like RNA from the virus that's expressing one protein, or what do you have any idea? I'm coming from I'm from a virus background, so it's really.
So in case of the replication deficient adviruses, they can exist, the DNA from those can exist
Oh, it's
epizomally.
DNA,
Can exist epizomally in macrophages or some other cell types for quite some time. And this was actually shown before we were looking at it in a TRM context where people just like encoded GFP in these vectors, and they could find it like 3 or 4 months later within these alveolar macrophages that were still glowing green, 4 months after adenoviral immunization. In relation to some of the influenza viruses, we think part of the reason they decline is that they don't hold those antigen depots very well in that case. And so that's why the TRM decline. I think it does bring up an interesting question, though, kind of coming back to what Bruce mentioned, which is we don't know anything about long-term durability of these cells in humans, right? And I don't know how well our animal models recapitulate humans or not. And of course, it's a super complicated and invasive thing to do in humans, right? Because you'd need repeated bronchoscopies, over many, many months to try to get an idea of durability of these cells in people. But I still think it's a really interesting and important question.
Yeah, this also gets to the Edward Nirenberg's question about, do we know anything about the persistence of upper respiratory tract TRMs? Yeah, for what it's worth, even antibacterial TRMs don't seem to last long in the lung.
So yeah, I'm glad you asked that. We actually see that the upper respiratory tract within the nasal cavity, those TRM do persist with all the vectors that we used. And this has also been confirmed by some other groups. So right now, we're not really sure why or what mechanisms, what signals in the nasal cavity are allowing those TRM to persist long term, but that is something that we're looking into.
Are the viruses also persisting in the nasal? I mean, you said if the viral vector persists, that can be one reason. So
right. Even with our influenza virus vectors that we use, they persist over time. And so the antigen in those cases, as far as we know, is being cleared.
Wait, so it persists in the nasal, or it's being cleared in the nasal mucosa?
Being cleared in the nasal mucosa.
Okay. Huh. So it's some other mechanism whereby they're persisting.
Yes.
Oh. Huh. Wow. It's interesting.
Yeah, we had a while ago, and this is a little, but we generate TRMs with the intramuscular vaccine or in mice. So presumably there's no antigen in the nose, and those TRMs persisted for, we measured from 6 months, and they were, at least in this mouse, but they were there for 6 months.
Yeah. And we see something similar with the IP priming. So I mentioned it is significantly less numbers of TRM in the nose, at least when we did it compared to intranasal, but they are still there. We can detect them. in that case. So those do persist, but the lung is different for reasons we're not exactly clear about. And other than the adenoviral vector, we've never seen any other scenarios where we can get TRM to persist in the lung in our mouse models, well beyond like 6 months.
Yeah, I was going to put out a hypothesis out there because I was talking to Dave about this once about why the lung TRMs don't persist. And I'd be happy to find collaborations to do this because I don't know how to do this. But I mean, the lung as an organ also expands and contracts all the time, right? So I almost also wonder if the constant bellowing action of the lung expanding and relaxing pushes the lymphatics and immune cells out of the lung into the lymphatics and then makes them leave. Unless there is an antigen in the tissue that keeps the T cells around. And I don't see a way to really stop a lung from expanding and relaxing and really answering that question. But if anyone has ideas, happy to
There's a
collaborate.
way to do that, Annika. You probably don't want to, though. Yeah. So one possibility with the lung, and this is kind of going back to some stuff we published, I think in 2019 or 2020. So we've always looked at TRM in the lung separated by compartments. So we have cells in the airways that we can recover by lavage and cells within the parenchyma tissue. And there's a chemokine receptor, CXCR6, that's often associated with TRM in lots of different tissues. But in the lung, the ligand for that chemokine receptor, CXCL16, is highly expressed by alveolar macrophages, but mostly by epithelium lining the large airways. And so what we proposed in another paper, or what we were able to show, is that if that interaction actually helps draw cells from the parenchyma to the airways, draws those TRM there in a resting state. And if you block it, you block this kind of sort of, I'll call it a low steady state movement of cells from the parenchyma to the airways. But once cells get to the airways, it's a pretty nutrient poor environment, and the TRM, once they're there, don't last very long. And we can see this with like intratracheal transfers and some other approaches. So it becomes a little bit of we think of it as almost like a conveyor belt. So cells from the parenchyma are being pulled out to the airways, and these cells are dying off, and more cells are being pulled from the parenchyma and into the airways, and they're dying off. And so the net result of all that over months is you're slowly depleting all of the TRM in these various sites in the lung. And so potentially that interaction is what's causing the gradual decline of those cells over time. They're being naturally pulled into the airway spaces, and they're just not surviving very long once they get there. And so by the 6 months after the fact, you've now essentially pulled out all the TRM from the parenchyma, they've gone into the airways, and then they've died off. And so the end result is no TRM anywhere. And we haven't nailed that down in any way, but I think that the data we have at least suggests that's a possibility for what's happening in the lung.
Yeah. I see Ellen has a comment. I'll have Ellen ask that comment if there's one.
Oh, no, I just think it's very interesting. Do you think they're getting pulled in the airway because so they have a better chance of encountering the antigen? So that's the adaptive value of having the lung cells make the ligand for that chemokine receptor?
Maybe. I mean, I would like to think that seems like it would be a reasonable evolutionary way to look at it. Getting the cells getting the TRM to closely associate with the cells that would become infected with the viruses they're specific for. I mean, if TRMs had a brain, I think that's what they would naturally do. So it seems like an elegant way of looking at that. But at the same time, for viruses that, as any population that's exposed to repeatedly over years, the idea of depleting or losing preformed TRM to some of those viruses may not be the smartest thing ever. And so I don't know how to kind of, balance those two ideas. But it could be something like that it's an effort to closely associate the TRM with the epithelium.
Where are they in the nasal passages, in the nasal mucosa? Where do they reside, these TRMs?
That's a great question. That's something we're working on right now. And I would tell you the answer if I knew it, and we don't exactly yet. We know some are associated with the epithelium. Whether their distribution differs based on the route of priming is something we're working on chasing down at the moment. And could that impact their protective efficacy based on how they're distributed throughout the nasal tract or the nasal cavity? We just don't have the answers yet.
Hmm.
In humans, we get plenty of CD69, CD103 positive, CD8, assuming TRMs on a nasal swab. So
Yeah.
we've studied them right off of nasal swabs.
Wow. Nasal swab or an NP swab?
Well, mid-turbinate.
Still, yeah, it makes a difference, right? So that's
It does. No, yeah. Well, yeah. So NP.
Yeah. So I mean, it would be really interesting to be able to longitudinally track like some antigen specific populations over time in that kind of scenario that Bruce is talking about, just to look at things like durability of a specific population over many months within the nasal cavity. Yeah.
How about mucosal antibodies? I don't know if you have any sense of how the kinetics, in the mouse model, how the kinetics are going, like if with a typical, like a flu infection or Sende, whichever model you use.
So we honestly haven't looked at these models to any great extent. So I actually don't know that answer. That's one of the areas that we're pushing toward a little bit with the transmission model in general is trying to focus a little bit more on mucosal antibody, but we don't know anything about durability yet of mucosal antibody here, whether it's IgG or IgA, but I think those are all really going to be interesting questions to chase down.
There's not that much data. It's hard to find data on the durability of mucosal antibodies.
It is.
Surprisingly.
Which is why I think, we're really excited, although it's not, kind of our wheelhouse, but I think we've got a neat system to play with to not only look at amounts of mucosal antibody, but how it might correlate with protective immunity in this case.
Okay, with time constraints, I'm unfortunately going to have to pull the plug on this discussion, but I'd be more than happy to set up a side Zoom meeting, and I'd love to be a fly on the wall and listen to all these questions and discussions. But given that it's a little over 3, I would like to thank Ms. Mikulec and Dr. Kohlmeier for participating and for the excellent presentation of a fascinating paper. I'd like to thank the audience member for the questions that they asked, and if some went unanswered, please feel free to reach out to the discussants or the speakers by email. I'd also like to thank our discussants, Dr. Foxman and Dr. Morales Nebreda, and all of you that joined and submitted questions. And for the attendees here, if there are any papers you think you would like to have presented by the authors via webinar, please contact us, and we'd be more than happy to host your pick of the journal club paper for the next session. And with that, we'll happily conclude our webinar. We'll see you in the next session, and I hope to stay in touch with all of y'all.
Thank you.
Great.
Thank you.
Thank you.
Congratulations again on the paper.
Thank you.
Huh? Oh, you.
Yeah. Bye. Thanks, everybody.
Take care. Bye-bye.